Um, well, thank you everyone for coming, and I hope that you have a wonderful time learning about the research that's ongoing here at CMS. Just a couple reminders. Please, when you're on the hallway, do try and stay quiet. There's people working in those offices, so they did ask us to remind you guys that. And I also wanted to remind a couple of different things from this year. This year, the presentations will not be judged, but we do have feedback forms available for those who want to give feedback. The blank ones are right there by the table, and then just put the completed ones on a tray right behind it. If you're attending virtually, I submitted the feedback forms in the chat on Teams. So just grab them from there and then email them directly to the speaker. I did want to remind, please keep the comments constructive. Do not comment anything that you would not like to receive as a comment. And for our raffles this year, they are based on attendance. So for every presentation that you attend, you get a raffle ticket. They're also by the door. So at every break, just come and grab how many tickets of how many presentations you've attended. And then without further ado, Delfina will introduce our CMS alumni speaker, and enjoy.
It is my pleasure to introduce the alumna, Kara. She received a BS in biology from Hope College in Michigan. Then she received her PhD here at CMS in 2013 from Dr. Ernst Peebles' lab. She has worked for the Florida Fish and Wildlife Conservation Commission's Fish and Wildlife Research Institute since 2013 and currently leads the Coastal Wetlands Research Group. She conducts research, mapping, and monitoring in salt marshes, mangroves, and oyster reefs across Florida. Today, she will give us a talk about her work to commence GSS. So, welcome.
Well, thank you for the invitation to be here. I'm always really excited to come back to the College of Marine Science. So, would you like the lights turned off up here? Is that helpful at all to see in the back?
Yeah, we can start the first one. The other one.
Perfect. All right, so this is not going to be a traditional scientific presentation. And instead, I was asked to present a little bit about my story in science and if there's any advice to pass on to graduate students here. So this is more like a science biography. So I completed my undergraduate at Hope College in Michigan. I'm from Grand Rapids, Michigan. So I majored in biology, minored in music and chemistry. Holland, Michigan really is as Dutch as that photo looks like. On the side, I was actually a tour guide in that windmill, wooden shoes and Dutch outfit and all in college. It was adorable. But during my time at Hope College, I did do some undergraduate research. It was a small school, so it was nice that I could get to know the professors there. And I did some research on the impacts of an endophytic fungus that we grew within grasses and then fed to caterpillars that we then fed to parasitoid wasps that would lay their eggs on these caterpillars and eat them alive. And we were studying if the fungus impacted the survival of the wasps. So it was a trophic level study. And that actually did end up getting published. So that was my very first publication as an undergrad, which was exciting at the time. And during that time as an undergrad, I also studied abroad in New Zealand and spent a semester there. So there was no marine biology offerings at my school in Michigan, but I was able to take marine biology in New Zealand, and that did kindle an interest in marine science at that time. So when I graduated undergrad, I had no idea what I was going to do. I thought about grad school, but it wasn't necessarily a dedicated course, so I decided to apply to a whole bunch of internships around the US. And I ended up at an internship at Mote Marine Lab down in Sarasota. And I started off, as many people start off in marine science, with charismatic megafauna, with the manatees. So I started in the photo identification lab down at Mote Marine Lab. This was a full-time unpaid internship. So I was also waiting tables at night and scooping ice cream on the weekends in order to fund my internship down at Mote Marine. And during the days at Mote, we were photographing manatees because so many of them have been hit by boats. They have a lot of scars on them, and each manatee has their own name and number and classification system and photos, and they can track the actions of individual manatees around Florida. So we were photographing manatees and then inputting all of their paper records into the computer was the job of the interns, of typing in all of these paper records into Excel forms. I stuck around for the next semester and then did some research in red tide. So the natural progression from charismatic megafauna to phytoplankton, as a lot of us do in marine science. And at Mote Marine Lab, that was when I learned about the College of Marine Science here at USF, because several of the scientists had attended here, and I applied here along with several other schools, but USF CMS definitely stood out as a great graduate opportunity. And I attended here from 2008 to 2013 in the lab of Ernst Peebles. But I really adopted the Paleo Lab as my home, so I spent a lot of time in the Paleo Lab and working with the other professors there in the Paleo Lab as well, and completed a lot of analyses on the mass spectrometer in the Paleo Lab. So I was fortunate enough to come in on an active project that Ernst had already had funded, so it was good to be able to hit the ground running with research. And this project was in the Caloosahatchee River estuary, where we were sampling 14 locations up the river at night. So 2 nights a month, we would go down to the Caloosahatchee and start the boat up at sunset. And we were lowering this setup that Ernst had designed with a YSI sonde for measuring water quality, diving pan fluorometer for measuring fluorescence on the bottom of the river, triplet fluorometer for more fluorescence measurements, and a sediment sampler for picking up surficial diatoms and sediments, and also pairing this with a zooplankton collection on another boat, partnering with students from FGCU. And this led to a study focusing on the cyclical nature of benthic dominance within the ecosystem, and then phytoplankton blooms and settling on the bottom of the river, and how this this turnover between benthic dominance and phytoplankton dominance within the system. So continuing in the brief history, another study that I worked on, so that previous slide was with Greg Ellis, for those that remember Greg, and this is now Elon Malkin, for those that remember Elon. We built this ladder of glass that we suspended right off the docks here in Bayboro Harbor. And this study was looking at how light impacts photosynthetic fractionation of algae that grew on the glass. So we lowered that same suite of instruments down through the water column several times a week through the course of a summer, and collected a whole suite of data on nutrients and harvested the algae every couple days. And we were able to look at what happens in the harbor over the course of the summer. Breakfast is ready. And so we could see with these pulse rain events that we had this low salinity surface layer that would come into the harbor that had a whole lot of nutrients, especially at the beginning of the rainy season, high amounts of CDOM, and then it kind of tapers off towards the end of the rainy season where you're not seeing as much nutrients coming into the harbor. And we could, using all this data and more that I'm not showing here, model the factors that are influencing fractionation of the algae that was growing on these plates. So the work on stable isotopes continued with an ISOscape project. So this was in partnership with FWC because they have research cruises with the CMAP program where they go out on the West Florida shelf to randomized locations here, and they conduct fish trawls here and collect everything that comes up in the nets, sort it all by species, count everything, measure it all, and it's fisheries independent monitoring that they're conducting here. But I was fortunate enough to tag along on these cruises, and I was collecting samples of this fish muscle tissue over this huge geographic area. So myself and a couple other students and interns worked on these cruises to collect an enormous data set that we could then analyze on the mass spec here at CMS for carbon and nitrogen stable isotopes, and creates isoscapes. So these are landscapes showing isotopic gradients within the Gulf of Mexico. So here I'm just showing nitrogen, how we have influence from the Mississippi River, and there's a gradient along the West Florida shelf for isotopes. And it's fairly stable from year to year and seasonally as well. So that also led to some modeling exercises using satellite data to model isotopic composition on the West Florida shelf. During my time here, it was also the Deepwater Horizon oil spill. So I didn't directly work on these projects, but for those of you that were here at the time, it kind of turned the school upside down, and that everyone was trying to figure out how we were going to best study the impacts of the oil spill. And being situated right here, it was important for the school to respond. And the impacts of the oil spill have continued to reverberate through the marine science community in the funding that it's provided. And honestly, a lot of the projects that I'm working on now at FWC were funded by oil spill funding as a result of the settlements that are working on a huge array of projects. So this was an important milestone, both unfortunately for the event, but it's also had large impacts with research thereafter. So during my time here, I also did spend a fair amount of time teaching. So I don't know if this is still very common, but when I was here, a lot of the grad students in the summer, we would teach some summer courses through Duke TIP, which is the Duke Talent Identification Program. And Johns Hopkins University has the Center for Talented Youth. These are summer courses for middle school students and high school students that are gifted and talented that they run across the country. Any students here, have you worked for Duke TIP or CTY? No? All right. So just know that these exist. They're great teaching opportunities. And every summer they hire graduate students oftentimes to teach these courses, and they have courses focused on marine science and oceanography. They're located all over the US. Some are at the Duke campus, but some are at their field labs. They've had classes located at New College and Eckerd College. Sometimes you don't even have to travel far. But it was a great opportunity to be exposed to teaching and developing a curriculum through these courses. They're really intense. It's like 9 hours of class a day. So you don't sleep much between all the preparation and grading during these courses. But it was a really good experience and exposure to teaching and developing your own course content. So I often taught in the summer with those labs, and then also TA'd over at USF St. Pete with their biology labs, which I think is still pretty common today for students to go teach the undergraduates there. So after graduating, I ran away from science for 3 months and went to Spain, and it was amazing. So I did some Spanish immersion, and then Reggie, my husband, and I would hike the Camino de Santiago. So this is also something I highly recommend if you ever have a break in life, that you can just take the opportunity to travel, because it does get harder as life goes on to take that break. But then I came back and had to figure out what to do with life again. So that's always kind of after you graduate, things are very uncertain, and that can be really stressful about figuring out what happens next and where are you going to find employment. So I continued teaching for a little bit to fill the gap in employment. I taught again for Johns Hopkins CTY up in Rhode Island. I was an adjunct professor at St. Pete College teaching oceanography, and I adjuncted at USF St. Pete teaching biology there. And then that fall, I was hired at FWC. So during this time I was teaching, I was also applying to every job that I could find in the area, because I had set up roots in the area, and my husband's job is in the area. And so I was trying to stay locally, and you have to either be flexible with location or job type. So you do need to have some flexibility with one aspect or the other. So I was applying to everything that I could find in the area. And I did get hired at FWC in their brand new coastal wetlands research program. So that was part-time at first. It was a brand new program, and there wasn't funding for me to be full-time. And so I continued teaching adjunct in order to still be working full-time. So I was back and forth between FWC and USF St. Pete teaching. And I continued teaching for a while until I had kids and it got too much. But I did continue teaching because I enjoyed it. So the adjuncting, it is nice teaching experience. It is not nice if you're trying to do it full time, because it is very, very low pay. And really adjuncting is a well-known issue within the university system about how low adjuncts are paid versus the demands of the job. I think at one point that fall, when I was adjuncting at so many classes, I calculated my rate per hour was around $7 an hour. Like, it's very low considering the time that you spend building lectures and grading. So it's definitely a problem within the university system. So it's fine if you're just doing one class on the side of your real job, but it's not something that's sustainable as a full-time job. So now I've been with the Coastal Wetlands Research Group for almost 10 years now. So it was kind of exciting getting in on the ground floor of this program because it was brand new within a year that I started. The research projects that we do, they're all grant funded. So I do write a lot of grants, and over time I was able to become a full-time position, and we eventually were able to hire more full-time people within the group. So this group was led by Ryan Moyer, for those that you may know him. He left FWC last year, and now I lead the Coastal Wetlands Research Group. So now I have 3 other full-time staff and 2 part-time staff within the research group. We do take on a lot of interns, so we try to always have 1 or 2 undergraduate or recent bachelor's students or recent bachelor's graduates working within the lab. And that's been a really nice component of the lab, as well as mentoring these students over the years and seeing where they all go in their scientific careers. So within the lab, we conduct research. We do research projects, mapping and monitoring in mangroves, salt marshes, and oyster reefs. So these are very different habitats than what I worked on, well, not too far away. And we're still looking at estuaries here, but I had not studied mangroves before I started my job at FWC, and now my life is all mangroves and salt marshes and oyster reefs. So it was a slight shift in focus, but a lot of the skills learned here are directly transferable even if you shift the research topic. It's still all science and understanding how to do science. So we are based in St. Pete, right there next door, but we operate across Florida. So this does involve a fair amount of travel around Florida to conduct all these research projects. So one of the projects that we work on is looking at blue carbon. So blue carbon, it is just a term for the carbon that's stored in coastal and marine ecosystems. So plants photosynthesize, they're taking up carbon dioxide, they store it as organic matter within the vegetation. But then when that vegetation dies, it also accumulates underground because the soil in coastal wetlands, it is anoxic. And so it doesn't decompose very rapidly. So organic matter can stay there stored for hundreds to even thousands of years in these layers of peat deposits that can be pretty deep. They can be a couple meters deep in Florida. They're a bit shallower up here in Tampa Bay, but if you go further south, there's some really thick peat layers down there. And these carbon stocks are very important when we think in terms of global warming and climate change. We want these ecosystems to remain stable, because if they die and the peat starts decomposing, that's going to release CO2 back up to the atmosphere. So we want to protect the ecosystems that are remaining and also encourage restoration to add additional area to these habitats. So we do spend a fair amount of time quantifying the amount of carbon stored in these ecosystems by measuring the vegetation and collecting soil cores, which we then analyze through a loss on ignition process, where we dry out a certain amount of sample and then combust it at a high temperature to burn off the organic matter. And the weight lost will tell us how much organic matter is stored in the sediment. And then from there you can calculate how much of that organic matter was carbon. So we do a lot of carbon calculations, and recently we've been working on a carbon model for all of Southwest Florida coastal wetlands. So this is a model where we used all of the data that we've collected over the years and also pooling other published data sets to model carbon density within the soil, and then I developed a separate model for the vegetation above ground. And then I used a suite of predictors from satellite data in order to select the best predictors for modeling this carbon density, and then extrapolate this across Southwest Florida to model how much carbon is stored within the coastal wetlands. And a lot is stored along the coast here, which unfortunately is the most vulnerable for sea level rise and hurricanes. They get hit pretty hard along the coast here. So this project has been in collaboration with USF. So Brad Rosenheim has been working on this project, as has Donnie Smoke over at USF St. Pete. We have other collaborators at UCF and Eastern Carolina University. And it's also partly led to another project that we just got funded, where Brad Rosenheim is the leader on this project, where we are looking at carbon sequestration in restored coastal wetlands. So these are sites where they just scrape down the bare earth and then either planted mangroves or let them naturally recruit. And these sites, they're some of the oldest coastal wetland restoration sites in Florida. So they're 20, 30, and 40-year-old sites. So we're going to go back to them and see how much carbon has been stored there over time, both above ground and below ground, and see what elevations are best for storing carbon, what nutrient conditions are best, what inundation conditions are best to help improve future restoration projects in mangroves. Another restoration project that we're looking at, this is in Jensen Beach. This is a site that is entirely impounded, so it's surrounded by a berm here. There were some tidal connections with culverts through the berm, but after Hurricane Irma, there was standing water at this site for several weeks. And mangroves, they are fine with being covered with water at low tide, but they like to be exposed to air at, they're fine with being covered at high tide, but they need to be exposed at low tide so they can have air exchange with their roots. If they are constantly submerged, they'll die. And so there's a 50-acre mortality event here after Hurricane Irma due to standing water. So this is an active restoration site between FWC and the county there, and we're monitoring the forest as it improves over time as they're installing these culverts to help improve the hydrology. And so we have water level loggers out there. We're monitoring the flow, how it improves with all of the additions of the culverts that they're adding there, and tracking how vegetation is growing back in this region. Some other projects we work on include collaboration with the Tampa Bay Estuary Program and their critical coastal habitat assessment. This is long-term monitoring that we have transects established all across Tampa Bay, where we're tracking how vegetation is shifting inland as sea levels rise. So mangroves are shifting inland onto salt marshes, and salt marsh vegetation is going inland as well. So this is a long-term project where every 5 years we go back and see how the vegetation is shifting. And we've also spent a lot of time looking at damage caused by hurricanes in mangroves. So we have studies going down in the Keys and 10,000 Islands looking at the impacts from Irma, and then also just started studies down in Charlotte Harbor looking at impacts from Hurricane Ian and what influences the rate of recovery of mangroves. My group also runs the Coastal Habitats Integrated Mapping and Monitoring Program, or CHIMP for short is our acronym. So this is a program where we're compiling mapping and monitoring data for coastal wetlands across Florida. We have a statewide report, we hold workshops, we have an email listserv where we're constantly circulating publications on mangroves and salt marshes. And this proved to be pretty popular among the research community, and they requested that we do the same but for oysters. So now we also run the Oyster Integrated Mapping and Monitoring Program, which is WIMP or OIMP for short. We're great with our acronyms here. And this one is focused on oysters. And this was the first time that we were able to compile a statewide map for oysters in Florida was through this program. We didn't have a statewide map before. We didn't know how many oysters were out there. And we've also located gaps now for where we need to continue mapping for oysters because they're not all mapped out there. So we're conducting mapping efforts in collaboration with the Oyster Research Program at FWC as well, because there's a dedicated oyster group as well. So we hold a lot of workshops. About every year and a half, we have a workshop focused on either oysters or coastal wetlands, and we're filling in a lot of gaps in maps along the way. So looking back. If there's any pieces of wisdom that I might pass on, topics of research change over time, but skills that you learn to do science are very broadly applicable. If you learn how to use R, you can apply R to anything. And yes, it's true. Being good at coding really just means you're good at Googling and figuring out each step of the way of how you do R. Learning how to make your own maps in ArcGIS or QGIS, if you do open source, that's really important and very transferable. Scientific writing applies everywhere. Being a good writer and being meticulous with details is applicable everywhere. And just general knowledge of experimental design is important. So when people apply FWC, generally for each job posting I've had, I receive an average of 100 applicants for each job posting. Only about 25% have a master's degree or higher, so there's always the bonus right there. A lot of them are recent undergraduates. But I look for just general skill sets that we need of knowing coding. If you know ArcGIS, that's a big benefit. Scientific writing, that it doesn't matter so much exactly the topic you were focused on, but if you have these skill sets, you can apply them anywhere. If you know how to think like a scientist, that's what we need. In addition to being willing to hike through mangroves with a huge backpack in 100-degree heat for 12 hours a day. We also need that, but that's hard to put on a resume. You really never know until they come out into the field how people handle that. But being willing to put up with tough field conditions is another important part of marine science. And graduate school teaches you how to teach yourself. That is important because you are constantly encountering new topics through your entire career, and you need to be able to figure out through the primary literature how you do things, because there will always be new topics throughout your life. And so teaching yourself is important, and independent learning and seeking out help when you need to ask questions. So also looking back, networks matter a lot. You never know what something might lead to. I think all of the teaching jobs I got were just by word of mouth, that I knew that they even existed. And same goes with a lot of research collaborations. You're just talking to people, you never know where it's going to lead. So getting involved with things outside your own research is very important. And helping out grad students with their studies too, you never know what it's going to lead to. And it exposes you to a lot of different possibilities within research. Trying to figure out the next step is hard. And it's kind of impossible to figure it out until it happens, because you never know what kind of job you're going to get. And you might not even know that that kind of job exists until you are applying for it and in that position. So it is really hard trying to figure out the next step, but just know that in some ways it's impossible to predict. So just do the best at whatever you're currently doing. And then when it comes time to figure out that next step, just apply for everything and be open to a variety of options. Your personality has a large impact on the type of job that you will enjoy. I learned through teaching that I enjoy teaching on the side, but not as a full-time gig. I'm just too much of an introvert, and I need time where people aren't talking to me, where I can focus on my research. And I realized that was good for me to have that time where I could just focus on something. So trying out different types of things helps you learn that. Interacting with the public, that's a very special skill set and takes a very particular personality that enjoys doing that kind of thing. So personality is a really important thing here, and people that need interaction with people might not be happy coding on a computer all the time. So also think about your personality and what makes you content with your employment when you're looking at types of jobs. And enjoy grad school as much as you can. I know it's hard. But as you do progress through the impressed, oppressed, depressed, and then mostly pressed progression, I have found, and actually I remember when I was at a similar presentation in grad school that someone came back and said, enjoy the time that you have to focus on the science, because once you progress further in your career, the pace does pick up immensely, and so much of your time is focused on emails and meetings and paperwork and budgets and things that aren't science. So enjoy this time that you have that you can just dedicate to focusing on scientific concepts and delving really deeply into topics before you get distracted by 18 deadlines at a time. So even though it is very stressful at grad school, and I'm not diminishing that at all, but just recognize that it's also good in some ways to just focus on a certain select number of topics at once. Also, remember there's life outside of research. Life happens. You need to take the whole life picture into account. Be flexible on some aspects of your career type or location, and there's more than work out there. So whether it is having a family or having your fur baby family or traveling or spending time with friends. I think that there's definitely a workaholic nature within research, but it is important to save time for yourself in order to make it sustainable and not have burnout. So setting time aside for yourself is really important. And also, I mean, given how intense research careers can be, depression and anxiety are not uncommon. So take care of yourselves and look out for others and form a community where people are supportive of each other and not competitive with each other. And I think that really helps to build for the success of the research experience and also going in jobs outside of research, because a lot of people do continue on to jobs that are not research, and that is fine. There are lots of science careers out there that are not research, and you really never know if you might end up in one of them until after you've graduated and you see what fits you as a person. Oh, that I'm right at time here, so I don't know if I have time to take questions, but I can.
So was I really oppressive? No, I have a serious question.
Don't answer that.
Yeah, before she does, distract her. At the Jensen Beach impoundment, did you see subsidence associated with loss of organics?
So we don't have elevation measurements before, but with our current elevation measurements, it is lower within the dead stressed area.
Than other areas?
Than other areas, yes. And we found there's a similar area down in Alita River State Park in Miami that's almost the exact same setup. That dead mangrove zone also has lower elevation. So we don't have before after subsidence, but it is lower elevation.
Looks like it.
Yeah.
Very good presentation, by the way.
David?
Yeah, I was just going to say, excellent presentation, great messages. And a long time ago, you brought to our attention that some programs that you were applying to were concerned about the curriculum that we provided, and that they wanted you to have more biology courses to teach biology and so forth. So I checked around and I found there were some places like that and others that didn't care. And so I was just wondering if you've had any evolution thought about that and recommendation to the students here.
Yes, so that was in reference to, I was teaching biology at USF St. Pete, and I applied to a few biology teaching jobs, and they wanted a certain number of graduate credits in biology. And at CMS, it's very interdisciplinary, and you're taking courses in all different types of topics. And so I found I wasn't qualified to teach biology at some institutions. You could teach marine science or oceanography, but it's just something to keep in mind that, it's more common to have biology majors than it is to have marine science majors. So if you want to go into teaching, it's just something to keep in mind as you're selecting courses here, if you need that certain number of biology credits or not. And it is highly variable with institutions. Yes?
Why are you back here? Hi. So we teach a lot of mangrove kind of lessons for education and outreach activities. What are some things you would want us to share with young students?
Oh, one of the biggest reasons that we come up with for supporting mangroves is that they are essential fish habitat. So commercial fisheries, sport fisheries, when those fish are babies, they're living in mangroves and salt marshes. So even at FWC, that's the big justification for why our research program exists, is because it provides habitat. So for the fish species, bird species, invertebrates, and also protects our coastlines. It prevents erosion during hurricanes. It helps buffer storm surge. They help filter out nutrients. So probably the ecosystem services are important to emphasize. Yes.
Just curious on the when you're mapping the habitats and the ecosystem habitats, the oyster biomass, is seagrass included in that or not? Because it didn't seem to match.
So some seagrass efforts also map oysters. A lot of them don't. So, and then some mapping efforts are dedicated specifically to oysters. So it just depends. Like SwiftMud, they'll map seagrass and oysters together, but a lot of mapping efforts are just focused on one or the other.
Quick question about chimp. What kind of mapping is actually done in the chimp program? Is it bathymetric mapping or LiDAR mapping, or how's the mapping done? Like walking around? Or?
We draw from the Water Management District land use land cover maps, and that's just using aerial photography.
Aerial photography, okay.
Well, thank you. I want to keep you on schedules. I love you.
We're gonna take a 5-10 minute break, so we're
Sure.
so we're working back.
Kara, are you going to be around a little bit? Because I wanted to talk to you about science things.
I should have brought.
So then,
Okay.
I don't know if you guys are planning this, but you might like
you think it won?
But I think God said to use this.
That's what I was gonna use. Yeah, just because it like shows up really nice.
We'll see. I might
Oh,
also
yeah.
just be like in the upper right corner. Somebody else will. We'll see how it goes.
Okay.
I hate being tied to the podium.
Yeah.
How this goes. Do I have to? Everything is equal.
I got you. It's okay. It's okay. It's gonna be great. It's gonna be great. Yeah, just tell me. Alright, you ready? Okay, everybody, if we can get your attention.
Oh, there's a trick I showed you.
We're gonna start up again now, you guys. I don't know what's good. Yes. Okay.
That's what I did.
All right, so our next speaker is Olivia Blondheim. Olivia is a PhD student in Chris Stallings' lab, and today she'll be presenting on her work filtering friends, evaluating bivalve water filtration services. So,
Thank you so much. Well, good morning, everyone. It is such a pleasure to be here, and I'm so excited this morning to share with you a little bit about my proposed research looking at bivalve water filtration here in Tampa Bay. Dr. Archibald just gave us a fantastic talk talking all about our oysters and salt marshes and mangroves, and so I've been particularly interested in those ecosystems as well the past few months. And so what I'm going to be focusing on today is talking about oyster reefs and some of the ecosystem services they provide. Now, an oyster reef is like one up in the right-hand corner that you'll see, and they typically form in low-energy environments where oyster larvae or spat are able to settle on these hard substrates, and then over time build these oyster reef structures that you'll see right here. Now, they provide a lot of ecosystem services that are really important for us here in Tampa Bay, one of them being habitat for marine organisms. So they're important for a variety of fish species and also invertebrate species that settle on those reefs. They also are important for shoreline stabilization and erosion control, which if we think about some of the stronger storms and hurricanes that we experience here in the Tampa Bay area, oyster reefs can help to prevent some of the wave energy that impacts the shorelines. Also, historically around the world, oyster fisheries have been incredibly important, but we'll talk a little bit more about that in a bit. And lastly, they're really important for water filtration. Now, oysters are filter feeders, so they will filter feed phytoplankton, and in the process, they remove nitrogen and phosphorus and other nutrients from the water. So they're great for when you have a lot of phytoplankton in the water column for removing a lot of those extra nutrients from the water. Now, just to give you an idea of how important oysters are for water filtration, this gives you an example of how excellent they are at filtering the water. So in the left-hand tank, you can see all of these particles, sediment, nutrients in the water. And in the right tank, you can see when oysters have been introduced. Now, one individual oyster can filter up to 50 gallons of water a day. So if you think about all of these oyster reefs here in Tampa Bay, you can just imagine how much water they're actually filtering. But oysters are in big trouble. Around the world, oysters have been named as one of the world's most degraded marine ecosystems. And in some cases, they're more degraded than some of the other ecosystems we're more well aware of, such as tropical rainforests and coral reefs. Now, in this study that was conducted by Beck et al., they estimated that in the past 150 years, about 85% of our oyster reefs have declined globally. Now, you can see here on this map, the blue is representing areas that still have healthy, sustainable oyster populations, and the red is showing areas where it's poor or functionally extinct. So that's areas that more than 90% of those oysters have been lost. So you can see areas in North America, Europe, and Australia where there's been a lot of oyster fisheries over the years and some unsustainable harvesting techniques, those are areas where oyster reefs are really in trouble. Now thinking about how we can really help to restore some of these oyster reefs, there's lots of different methods, but one that has been of particular interest lately here in Tampa Bay is these vertical oyster gardens. Now this is where you have like this rope structure where oysters have been attached, and you can deploy these from piers or docks. And what they do is they help to restore some of that structure that these oysters provide, and also some of those water filtration sources or services. Now, I've been thinking a lot about oysters lately, and of course, how in decline they have been. So one thing that I was really thinking about is how does this really affect the water filtration services here in Tampa Bay and ultimately in other places around the world? And so something I'm really interested in is what are some of the factors affecting bivalve water filtration, and how can we better evaluate them? Now, oysters grow on a variety of different substrates. So like on those oyster reefs that you see up in the upper left-hand corner that we just talked about, but they also can grow on mangrove prop roots, on pilings and seawalls, so more artificial structures. And when you think about oyster substrates, something that was really interesting to me is how do water filtration services potentially vary based on these different substrates? I'm not the first person to think of this. A CMS alum, Dr. Michael Drexler, conducted a study back in 2008 and 2009, estimating a variety of different properties of oysters, including their sizes and densities on many different substrates. But two of the main ones he looked at were oyster reefs and mangroves. Now, one thing that he found that was particularly interesting was that on oyster reefs, the densities were much lower, but those oysters were also much bigger. And so there's some trade-offs here that we may be seeing where on mangrove roots, you may have a lot of oysters, but they're not as large, so they may not be clearing as much water. But on oyster reefs, you don't have quite as many. So there's some interesting things that we can think about here in terms of water filtration based on the substrate that they're on. So what I'm hoping to do with my first study is I would like to evaluate in situ the water filtration rates and capacity of the eastern oyster on different substrates. Now that sounds kind of challenging to do, but what we are planning to do is to select sites around Tampa Bay using a randomized design. And what we can do is pick different substrates like our control here, which doesn't have any oysters, an oyster reef, and mangrove prop roots. Now the cylinder that you're seeing represents a barrel that has the bottom and the top cut off. And so what we can do is we can go out to an oyster reef, place that barrel in the sediment, isolating those oysters and the water that's around them. And then what we can do is take an ambient measure of the phytoplankton that is there. And so over time, we can take samples from the water at set intervals and measure how those phytoplankton concentrations may change. And of course, applying this to all of these different substrates and seeing how they may differ between the substrates. Now, historically, you'd have to spend a lot of time under a microscope counting every individual phytoplankton cell, but luckily technology has advanced quite a bit since then. And so I'm planning to use a tool potentially like this, which is a planktoscope. These are really accessible, cheap tools that you can buy online and build yourself. And you can use an iPad with different programs to get these really nice high definition images of phytoplankton cells. So here's a couple of diatoms that have been highlighted with the program. But what I'm hoping to do is to be able to look at my phytoplankton samples through a tool like this to just get a better understanding of how those concentrations are changing over time. Now, again, going back to substrates, a lot of times we think of all these different factors like size and density that play into water filtration, but we don't really think too much about the actual substrate they're on and the orientation it's in. So something I'm also interested in looking at is how do oyster water filtration rates and capacity differ between oysters oriented horizontally, like on a reef, or vertically on mangrove roots or those vertical oyster gardens like I showed you a few slides ago. So what we can do is take these oysters back into the lab, and the cylinders here represent 250-gallon aquarium tanks that we find here in the aquarium room. And so these oysters are attached to PVC pipes, and we can orient them in different directions. So on the left side, vertically, like on a mangrove prop root, or horizontally, like a oyster reef. And then again, we can seed the water with a known set of phytoplankton cells. And then over time, we can again take those samples at set time intervals and get measures of how the phytoplankton concentrations may be changing over time. Now, one last thing that I'm very interested in is thinking again about water quality. And here in Tampa Bay, we're very interested in harmful algal blooms and how they're affecting our ecosystems here. Now, this is not just a topic that I'm interested in, but also a lot of aquaculturists and ecosystem managers are really interested to see if different bivalves can potentially help filter harmful algal blooms. Now, studies have shown that many oysters and different bivalves are able to actually harbor those toxins in their tissues.
And so we know it's not safe to eat the shellfish during these blooms, but there's not a lot of focus potentially on the water column and what's happening there. And so with my study, I'm hoping to use 3 different types of bivalves, so oysters, clams, and mussels. And what we would do is place them in different tanks. And again, using similar methods where we would seed the water with a known amount of phytoplankton, we can then measure over time that removal rate and see which bivalves are most effective at removing those phytoplankton concentrations. Now, something that we're hopefully going to be able to do with this is once we get those preliminary results, we can actually mix and match some of these different bivalves to see if there's optimal combinations. So when ecosystem managers are going out and actually placing clams and mussels in the field or oysters and mussels, they can figure out whether they should be planted together or in different groupings to maximize harmful algal bloom removal. And so this could be a great tool for restoration efforts moving forward. Now, one reason I'm particularly interested in this, as Dr. Radebaugh mentioned, a lot of ecosystems are starting to change here in Tampa, particularly with the vegetation. So mangroves are starting to shift into salt marshes, but they're also starting to shift into oyster reefs as well. And so a study recently conducted by Dr. Stephen Hesterberg found that 83% of oyster reefs without any initial mangrove cover fully converted to mangrove islands here in Tampa Bay. Now, on the left image is a aerial image of oyster reefs here in Tampa going back to 1938. And you can see those white circles are those oyster reefs. And the mangrove is, little mangrove patches right up in the middle part of this. So you can see there's not too many mangroves here. A few decades later, in 1995, you can see more mangroves have encroached into oyster reef habitat. And so they're starting to kind of take dominance there. And then lastly, in 2020, you can see one of those islands or oyster reefs has fully converted to a mangrove island, but the others are also well on their way to becoming more mangrove dominated. So here in Tampa Bay, we're expected to see quite a few changes in the vegetation that we're seeing and which is dominant. So it's really important for us to understand, as many of these oyster reefs are being replaced by mangroves, how may that have an effect on water filtration? So I'm hoping by my studies, we can help to really build up some of that baseline information we can use and then apply to something like this to get better predictions for how water filtration services may change here in the Tampa Bay area. So with that, I'd like to acknowledge the following people and thank you.
So if there's any questions, maybe if we can do student questions first and then any faculty.
So I saw your 3 maps,
Mhm.
and there seems to be a big difference between the second and third map.
Mhm.
So what in particular is causing that such a much bigger shift for the mangrove islands to take over the oyster reefs?
Yeah, absolutely. So from a lot of studies recently, particularly looking at mangroves and salt marsh, we know that mangroves are really kind of shifting towards the poles and kind of invading in these previously dominated areas like salt marsh and oyster reefs, because as our climate's changing, typically mangroves are pretty not very resilient to freezes and changes in rainfall patterns. So as climate change is making more and more air temperatures, fewer freezes, and just these different variations in rainfall patterns, mangroves are able to establish much easier in place, or are able to establish themselves in places that they haven't been established before. So, obviously in the first photo, it probably was, some of those changes were first kind of taking place, and it takes a while for some of these juvenile mangroves to actually establish themselves. But once they start growing, they can really start to kind of spread, and you're getting more of that seed propagation. And so it's much easier for them to kind of really take over areas. So I spent a lot of time this past summer up in Cedar Key, where you can see places that, previously were all salt marsh or just all mangrove. And then of course here in Tampa Bay, we're also, seeing that with the oyster reef. So yeah, it's usually just like the changes in some of our climatic patterns. And of course, once they're established, you have all those nice seedlings that can drop and start to establish as well.
Thank you.
Yes.
Thanks, Dr. Olivia. I had a question about your experimental design with the barrels.
Mm-hmm.
Like, what sort of dimensions are those, and like, how can you account for tidal changes or like damage from storms?
That is a really great question, and I'm still working out all of the details of that. So this summer we used, I forget the particular dimensions, but they're probably about, they're decent sized barrels. They're really large. And so historically, kind of the idea behind this came from when you're sampling invertebrates in salt marshes and oyster reefs, you use these nice large barrels to kind of isolate everything. And then you use like these huge like fire hose sized tubes to kind of remove a lot of those invertebrates in the water there. So that was kind of the idea behind the barrel, was we know that we can isolate invertebrates that way. And so, and just from experience using that, a lot of times the water gets trapped as well. So I'm hoping that with these barrels, we wouldn't be setting them out for days at a time. These would probably be within 24 to 48 hours, and then setting those like time intervals. So hopefully by that, we can kind of account for any storms. I'm not, with tidal changes, we still have to kind of work that out a bit. Hopefully there wouldn't be too much of a change if we're able to isolate it. But again, these are kind of prototype designs based on other methods that I've used using, sampling for invertebrates in the past.
Sure, yeah.
Yes.
Follow-on question. What was the control substrate? Is it hard bottom?
It'd probably be hard bottom, yes. So, and we'll do that somewhere adjacent to some of these habitats. That way you can really kind of get a measure between, if a mangrove crop roots are over here, so that way you're having similar phytoplankton species and concentrations ideally in that area. Yes.
So when you say that mangroves are replacing oyster reefs, are the oysters actually being lost, or are they still there and just growing amongst the roots instead of in true reef formation?
So from what I know, they're primarily kind of just like, so in some of the areas, yes, there still is that oyster like foundation there. But for the most part, once those roots kind of take over and they start to really establish, a lot of those oysters can no longer really stay in those areas. So you are still losing the oysters in those places. But again, that's another kind of thing that we're interested in is, are these oysters, other larvae starting to settle then on the roots, and are they replacing them in different ways? And so there's still a lot of unknowns in that, but yeah, you still have the substrate and some of the oysters there, but most of the time, a lot of them are being lost.
Okay, I think we're going to move on to the next one now, just to stay on schedule. So thank you so much, Olivia. So our next speaker is Caitlin Parente, and Caitlin is a master's student working with Kristen Buck and Maya Breitbart, and today she'll be presenting on her work on marine phages.
Good morning. Yes, I'm Caitlin Parente, co-advised by Dr. Maya Breitbart and Dr. Kristen Buck, and this is my work investigating marine phage as a potential form of dissolved iron in the ocean. Let's see. So iron limits primary productivity in roughly 50% of the surface ocean. Iron is a trace metal that's really important for a lot of biological processes like photosynthesis and nitrogen fixation, but iron has really low solubility in seawater, and it's highly likely to be complexed by organic ligands in the water column, so we don't see a lot of iron. And taking a look at this figure on the right, anything in blue is predominantly iron limited. So even among different community compositions of phytoplankton, you're seeing iron limitation as a driving factor. In stark contrast to our very limited iron in the ocean, viruses are everywhere. So viruses are our most abundant biological entity. There are 10 million viruses per 1 milliliter of seawater. Comparing bacteria and viruses, they're roughly at a 10-to-1 ratio, viruses to bacteria. Most viruses that we see in the surface seawater are bacteriophage, so they specifically infect bacteria. I'll call them phage from here on out. And so this is one example of a tailed phage, and what a phage does is it attaches to a bacteria cell surface, injects its viral DNA, and commands that bacteria cell to replicate its viral DNA. And so it makes all of these progeny phage until the cell can no longer hold them all. It lyses the cell or bursts that cell, and all of the progeny phage along with cell contents are released back into the water column. Because of this, viruses are considered a driver of biogeochemical cycles. So the pictures on the right, you can see viruses attaching to that cell surface. And then here at the bottom, this is that cell lysis, when all of those progeny phage are released, and in red is just one single phage. So viruses and iron are not often discussed together, but they do fall in the same size fraction in seawater. So we operationally define seawater size fractionation with filter sizes, and so we distinguish between dissolved, which is on this left a little more than half, and particulate with a 0.2 micron filter. So anything that passes through a 0.2 micron filter is going to be considered dissolved. That is the size fraction of seawater where phytoplankton are obtaining iron. And viruses also fall in that dissolved category. We further distinguish between soluble and colloidal with a 0.02 micron filter. But in general, viruses and iron are in this same size fraction and just not really considered together too much. But we have some insights from non-marine systems, and so people outside of our world have looked at viruses and iron. And we have seen through X-ray crystallography that this non-marine phage T4, which is a phage of E. coli, a very common bacteria, contains iron atoms in its tail fibers that are paired or coordinated through paired histidine residues, which is not that important to you probably, but they contain iron. So this is a big deal because if non-marine phage contain iron in their structures, then maybe marine phage do too. And so with this kind of hypothesis and thought, in 2019, someone went through the Terra Oceans metagenomes that were collected from the Terra Oceans expeditions and found these potential iron binding sites in 87% of those metagenomes. So there's a high likelihood that this could be worth exploring. And I think that gives us plenty of incentive to look into this, because if viruses contain iron in their structures in the ocean, then this could have significant implications for our understanding of iron biogeochemical cycling. So these lines of evidence that marine phage are overwhelmingly abundant in the ocean, they fit in the dissolved size fraction along with iron that phytoplankton are taking up. And then non-marine phage include iron in their tails. These lines of evidence laid the foundation for the Ferrogen horse hypothesis. Chelsea Bonnet-Chase, a former USF graduate student, and Maya and Kristen wrote this hypothesis paper, and so we think that phage could be comprising a significant portion of dissolved iron in the ocean if these things are true. So I'm going to walk you through the broader Ferrogen horse hypothesis, and then we'll go into my work a little bit more specifically. But this hypothesis says that phage that contain iron in their structures might be competing with siderophore-bound iron or other iron for iron uptake receptors on the cell surface. And so the Ferrogen horse is this kind of Trojan horse idea that the apparent gift of iron in these tail fibers is allowing viruses access to the cell that they might not otherwise have. So the phage are infecting the bacteria cell with their viral DNA. This is the part where they're going to command that bacteria cell to replicate their DNA. And another part of this hypothesis is that all of the iron contained within the progeny phage is originating from that bacteria cell. So they're using bacteria cell reserves in the production of progeny phage. And last, those phage are released back into the water column along with cell contents, and the amount of iron available for remineralization is diminished compared to if there wasn't iron in the structures of these phage. So I'm revisiting this hypothesis. Chelsea established methods for looking at that non-marine model system with E. coli and phage T4 when she was here, but I will be adapting those methods and applying them to a marine model system in order to answer the first critical question in starting to tackle this hypothesis, which is, do marine tailed phage contain iron in their structures? My hypothesis is that they do. I think that they include iron as part of their structures and consequently account for a portion of iron in seawater. And I also think that progeny phage are obtaining that iron from bacteria host cell reserves. So a broad method overview, I'm using Vibrio nitrogens as my model bacteria. It's a fast-growing marine bacteria that a lot of people around the world are starting to work with, which is exciting. But I'll be growing that bacteria on isotopically labeled iron media. I will then infect Vibrio with phage, or as my control, I'll lyse it with chloroform to burst the cells. I'll purify the phage that are produced from cell lysis, and then I will analyze those phage for their iron content using iron 57 as my tracer. I'm going to walk you through a little bit my methods charts. Follow the green boxes, and that'll give you kind of the gist of what I'd like you to know. But I'll be preparing my liquid cultures at the first part of this, and then I'll be growing my bacteria to their ideal time for phage infection. So I'm going to separate my media and inoculate that media with bacteria with these 4 treatment designations and growing them until they're ready to be infected by phage, at which point I'll divide all of those samples with their bacteria into triplicates. So I have triplicate samples of everything, and I'm going to make sure that all of the iron 57 that the bacteria have been growing on is contained within the bacteria cells. So I'll go through a couple different washing steps and use ironless media or media without the labeled iron. So that way all of my labeled iron is contained in the bacteria cells. And then I'll get to this step. So this is when I'm gonna add my phage to that phage infected treatment group. And then all of the other treatments will be lysed with chloroform. And so the lysis of chloroform will just burst the cells as the phage will burst the cells here. And then I will remove any remaining bacteria. So this is a couple days later, moving into just isolating my phage from these samples. I will purify the phage with a sucrose cushion. I'll remove any soluble iron or anything that might be remaining through dialysis. I will count the number of phage with staining. I'll stain them with cyber gold and use epifluorescence microscopy to count my number of phage produced. And then I'll find the iron concentration, specifically looking for iron 57 throughout the process with the ICP-MS. Specifically, I will be using a high resolution inductively coupled plasma mass spectrometer. Iron 57 is our tracer. We will follow it from the very beginning, making all of my reagents and ingredients through to the end. And with the ICP-MS, that'll give us iron concentrations, and we can find atoms. Paired with our phage counts from microscopy, I'll be able to calculate iron atoms per phage, which is an interesting finding. And so the project status, I know it is only 6 boxes. It took a long time to get these checked off. This has been a lot of method development, which has been really fun, but I've determined my media. I've established all of my methods for this marine model system, which is working a little bit differently than E. coli and T4. I will be doing everything trace metal clean, so that's a lot of acid cleaning and preparing my materials for each run. And then I have identified KVP40 as the first virus that I'm going to use. So we have 3 viruses in our lab that infect Vibrio nitrogens. KVP40 is the most similar to phage T4 of the ones we have, so we think it's our best bet. And then I have gone through and done an XR run, so ICP-MS. My first one was yesterday, and so I am going to be analyzing those samples and kind of establishing a preliminary baseline for the media I'm using and all of the steps of this process. And then I'll be starting full experiment runs in the next couple weeks. This picture on the right is a plaque assay. So all of the little dots on that plaque, that's a bacterial lawn that's been grown, and the little dots are where the virus has killed the bacteria and cleared it. So it's growing well. Conclusion, iron is critical for biological processes. We know that it's very important for photosynthesis and nitrogen fixation, but it's found in really low quantities in seawater due to its low solubility and its high likelihood to be complex. Viruses, on the other hand, are super abundant. They are everywhere, and they are drivers of biogeochemical cycles, but they haven't been looked at with iron specifically. So marine viruses might contain iron in their structures, and if they do, that could have significant implications for our understanding of iron biogeochemical cycling in the ocean. And this work will provide a foundation for other people to look into this issue and answer a question that is a really critical piece of the puzzle. So I want to say thank you to Salvo and Mackenzie for all of their endless help in the lab and troubleshooting. And then thank you to the fellowships and funding that allow me to do such exciting work with really brilliant people. Thank you guys for your time and attention. I'll take some questions.
Thanks, it's really interesting, so I have lots of thoughts. So the first thing was, like, I might have missed it, but do we know if viruses are in the colloidal phase? Like, we know that, like, which, can you separate them from the dissolved with a filter?
Typically colloidal is where they're going to be. So we will separate them with the filter and catch them on a 0.02 micron filter. And that's how we look at them for the fluorescence. Staining, they'll be caught on that filter and then can be stained and looked at under a microscope.
And the second point was, has anyone looked at how bioavailable that might be? Anything? I mean, I guess we're in early stages of this.
This is really early stages, so I don't think anyone has looked at that yet, but I would be interested to follow this work later and look at bioavailability with different, thinking of it maybe as a ligand and competitive exchange that would be a much later step, but we'll identify the iron first, hopefully.
What kind of saltwater will you be using?
I will be using VN media. So this VN just stands for Vibrio nitrogens, which is my bacteria. And so they've developed this media. It's defined, so I know all of the components, and that is a plus, but it uses all kinds of different salts, and I add trace elements to it. And then I just swapped the iron sulfate for labeled iron sulfate. So it's a little bit modified, but it'll be a media adapted for this bacteria.
In your methods, what was the difference between the bacteria control and the lysis control, or like the chloroform one?
Great question. So in my, I'm not going to click all the way back there, but I had 4 treatment designations. So I have the treatment that will receive phage and be lysed. I have the bacteria control, which is just bacteria and no phage. And so those bacteria cells will be lysed with chloroform to count for anything that's being released by the cell and not phage induced. And then the last 2, I'll have 2 blank media controls. One that I will just add chloroform to see if there's any effects from chloroform, and then one that won't receive anything the whole time. So that last one that is just media that'll be in the incubator for all 5 days, that one's just to make sure I'm not contaminating anything and that I'm doing everything clean on the micro side. Did I answer that? Yeah.
So you should be able to calculate the proportion of iron that's released as virus versus cell contents, right? Or
Yes,
lysis.
so that's what the bacterial control is for, to separate any iron that might just be released in bacteria cell parts following lysis compared to the one that is looking at the phage specifically.
Thank you so much, Caitlin. All right, and our next speaker today is Keith Keel, and Keith is a master's student working with Maya Breitbart, and today he'll be presenting on spawning dynamics of fishes on the West Florida shelf.
Hello everyone, I'm Keith, and as Catalina mentioned, I'll be presenting on some work I've been doing for my thesis, which is the assessing fall spawning dynamics of fishes on the West Florida shelf using DNA barcoding. So firstly, why study fish spawning? First of all, spawning areas can host a large amount of productivity and biomass in a relatively small area, especially for fish species that aggregate when they spawn. And this has conservation and management implications. And because of that, it's important for us to locate these spawning areas, especially for fish species that are important to us, such as ones that we fish for commercially or recreationally. Additionally, the early life stages of fishes are highly influenced by environmental variability, and so we're interested in seeing if fish egg distributions change over time in response to climate change, or changes in habitat quality, or even changes in fish populations. So we want to study fish spawning, and ideally we could go out and look for scenes like this, and we know which fish are spawning where, and that would be really fun. But unfortunately, the ocean's pretty big, and that's not really practical. So we have to do the less fun thing, but more practical thing, which is to use fish eggs as indicators of spawning location. And fish eggs are really great indicators of spawning location because they're passive and buoyant particles. So they can't behaviorally control their movement like fish larvae can, and they're also buoyant, so they typically rise to the upper layer of the water columns because of the lipids inside of them. They also develop very quickly in the warm waters of the Gulf of Mexico, typically hatching within 24 to 48 hours. So if we find a fish egg, we know that it's relatively close to that original spawning location. So the problem with fish eggs is that they're almost impossible to identify visually, but by using DNA barcoding, which I'll talk about a little bit later, we can determine which species of fish these eggs are coming from. And also, luckily for us, almost all commercially important fish species on the West Florida shelf are broadcast spawner. So they're kind of just spewing their eggs out into the environment, and we can just go and collect those. And so the objectives for this study are firstly to identify spawning hotspots for the fish species of interest on the West Florida shelf. And going forward in the future, we also want to further understand how environmental and geographical parameters are affecting the spawning dynamics of these fishes. So this presentation will primarily be focused on our first objective, which is to identify these spawning hotspots. And to accomplish this, fish eggs were collected in September of 2013, 14, and 19 and identified using DNA barcoding. So to collect all these fish eggs, we use what is called a continuous underway fish egg sampler, which we call the COOFs. And what this is essentially just a pump that's attached to the hull of a ship that sucks in water at a 3-meter depth and flows into a concentrator device, which is essentially a device that consists of an oscillating mesh net that's going to sort out fish egg-sized particles. And then this will flow to a mechanical sample collector, which is just a fancy way of saying a piece of PVC with some mesh glued onto it. And so the COOFs really excels at high-resolution spatial sampling because it can be used while the ship is moving, and you don't have to stop at each site. And so here's a picture of the COOFs aboard the ship, and a picture of a sample that we would get from one transect or site from the COOFs. And you can see this contains a mixture of zooplankton that is about fish egg size and fish eggs. And another nice thing about the COOFs is that we get these really clean samples that don't have a lot of gunk in it. If you ever looked through a sample you get from like a toad plankton net, you know that it can be really messy. So this saves us a lot of time. And so these samples were collected aboard NOAA CMAP cruises in mid to late September, about over a 2 to 3 week period in 2013, 2014, and 2019. And each of these sites corresponds to about a 10-kilometer transect. And so because we have all of those sites, we took a spatially balanced subset of 68 samples just to reduce overall time intensiveness of the lab processes. And so once we collect all those fish eggs, we can start the DNA barcoding process. And so this starts with a DNA extraction. So we'll take all of our fish eggs and put them individually into their own separate tube and fill that tube with a lysis buffer. We then manually pop those fish eggs using a toothpick. And what this does, yes, very tedious. And what this does, it bursts open those cells inside of the fish egg and releases that DNA for us to be able to use, and therefore extracting it. From there, we do a polymerase chain reaction, or PCR, and this will amplify our DNA, creating billions of copies, preparing it for sequencing. We send our amplified DNA out to a sequencing facility, and they're essentially just going to read those nucleotide bases in that DNA, and you can think of this as a code that is unique to a specific species. And so from there, we get our sequence, and we essentially just plug it into a reference database. We usually use the Barcode of Life database, or BOLD, and this has reference DNA sequences for over 24,000 fish species. We plug our sequence that we received from the sequencing facility into the database, and we look for a match, a sequence match that is set at a threshold of 97%. If we find a match, then we can successfully identify our fish egg. If not, then we use another database and look for a match in there. If there's no match in there either, this is pretty rare. There's usually a match in these reference databases, then we can't successfully identify our egg without further lab processing. Typically, our barcoding process is successful about 80% of the time, though we're not always able to get down to the species level. And typically those failures are happening at this PCR or sequencing step, and that can happen for a multitude of reasons. And so far we've barcoded over 4,000 fish eggs throughout the 3 years of sampling with a success rate of 81%, and we have found 84 unique fish species. So here's some of the species of interest we've encountered fish eggs from. We have demersal, reef-associated, and highly migratory pelagic fishes all represented in this study. And finding these species will further allow us to understand their spawning locations, timing, and environmental preferences. So here are some maps of where we found fish eggs for some of our species of interest. These maps include 2013, 14, and 19. It's also worth noting that in these maps, we're just looking at presence and absence. So at these different sites, we could have different number of fish eggs, but typically for our species of interest, we're only finding a few sometimes even just one fish egg at each site. And so for our yellow edge grouper, we have this interesting kind of concentrated spawning area in the southwestern portion of the shelf. For our red drum, we have seen spawning all the way up near Pensacola Beach, down south to the Keys, but we've only found 3 spawning events over the 3 years. For our king mackerel, we have a pretty wide north to south distribution, kind of along that mid-shelf region. The red snapper, pretty concentrated up near the panhandle region, but with some down about halfway down the shelf as well. Vermilion snapper also has a very wide distribution, kind of like the king mackerel along that mid-shelf region. And for the lane snapper, we typically see the spawning in the southern half of the shelf, with some exceptions. And it should also be noted that just because we didn't find fish eggs in a particular location doesn't mean that spawning doesn't occur there. We are just looking at about a 2 to 3 week period in September over these 3 years. And so we can also look across years. So here we have red snapper spawning that we found in 2013, 14, and 19 separated. And you can see 2013 and 2014 kind of look similar. Most of it's concentrated up near the panhandle, but in 2019 we do see not only more spawning events, but also a more southerly distribution. And so while we're mostly interested in those commercially and recreationally important fishes, the vast majority of the fish eggs we collect are from these groups right here. So we have scad from the Decapterus genus, the lizardfish family, sea robins from the Periodonatus genus, and sand flounders. And so while these species are of little commercial importance, understanding their spawning dynamics is still important for us to understand the full community ecology of the West Florida shelf. And by far the most abundant species, or group of species, was the scad from the Decapterus genus. And they were actually so abundant that you can see our cruise tracks on this map. They were almost found at every site. And so going forward, we'd also like to include some hindcast models of oceanic currents to kind of more accurately estimate those original spawning locations, especially for the fish species we're really interested in, like the yellow edge grouper and the red snapper. And so, for example, if we find a fish egg here, that original spawning location could be in a pretty wide range, because even if this egg is only 24 or 48 hours old, those currents can have a substantial impact in that amount of time. So if we knew where the currents were coming from, we could at least better estimate that original spawning location. I also plan to use some multivariate and spatial analyses with some environmental variables that were collected during the cruises to further understand how environmental factors relate to fish spawning. Thank you. I would expect them to be a lot different, just because different fish species have different timings when they spawn. Some are spring spawners, some are summer spawners, some are winter spawners. In this case, we're capturing fish that are typically spawning in that kind of late fall, early fall, late summer time period. So yeah, it'd be very different. And in the future, this project is getting bigger, and we will be sampling different seasons.
Yet another follow-up question. Is there a paper showing what is the likelihood of capturing spawning in those 2 to 3 weeks in September? Are there numbers specifically that show that, yeah, you should capture it?
Like number of fish eggs?
Just the likelihood to capture the spawning.
Yeah, we do have papers that document what time period these fishes are typically spawning in. So we kind of do have predictions about which fish we're going to see.
Is that like the maximum likelihood time of year?
So this time period was not really targeted at any sort of specific fish, just kind of in general those fish spawning in that time of year. Yes, Brad.
Could it be that you see fewer sites with commercially viable species because they're commercially viable?
Well, I don't think that's particularly why. I mean, I guess they have, less biomass compared to something like the scad, which is a forage fish. There's just more of them, so you typically see more spawning. But I don't know if you could really say that you see less because they're being overfished. I mean, if something's overfished, then sure, you'd expect to see less spawning as well.
So I was just curious about the track design over the 3 years. It looked like the first year the captain was drinking, and in the other years was that intentional to have these different? Because for temporal comparison, how will you work with that?
So I was fortunate enough to go on one of these CMAP cruises with Noah, and sometimes stuff on the ship breaks and timing shrinks, so you have to sample less sites, and they choose strategically which sites they want to choose. Sometimes a hurricane comes into the Gulf and you have to dodge that. So yeah, it's more so for practical reasons than scientific reasons. They do have a set grid of sites they do sample.
Oh, I see.
Uh-huh.
And more so than drinking.
Yeah.
Yeah. More so.
And you don't see any menhaden in this?
I don't think we've encountered any menhaden. No.
Interesting.
Maybe a different time of year.
Yeah.
They're in higher productivity areas.
Tim?
Thanks, that was a really great talk. You started talking a bit about, and I'm going to be dangerously busy growing up with you. But I mean, I was thinking, like you were saying, the loop current moves very fast, the hurricane moves very fast. Is there any thought that different eggs, because of their morphology, might be spread out more widely, or someone's bigger and settled near the location or anything like that?
Yeah, definitely. They do have different sizes, so that would affect how they're affected by currents and stuff. And also, they do have different buoyancies. So we're adding an additional study to see if eggs have different certain levels that they rise to in the water column and stuff. So yeah, that definitely could play a factor.
Thanks so much, Keith. And we have one last speaker this morning before our mid-morning break.
You don't have to read the title.
Yeah, I won't. So Louise is a PhD student working with Yongyang Liu and Bob Weisberg. And today he'll be presenting on ocean atmosphere heat exchange on the West Florida shelf.
Hello, my name is Luis Sorinas. I'm a 5th years PhD. I don't know, I've been here a while. With Bob Weisberg and Yongyang Liu. Oh boy, it's a long title, so I'm going to summarize that for you. I'm going to be talking to you guys about heat flux in water column temperature seasonal cycles on the West Florida shelf, looking through like using 2 long-term mooring buoys. So what's the motivation for this? What do we care? As we know, the ocean is a major player in the air climate system, and a lot of that is due to the capacity the ocean has of store heat and distribute heat around the world. And we feel that particularly in coastal areas, as the ocean plays like a regulator role. A lot of that regulation of climate due to the releasing heat during the winters and gaining heat during the summers happen through the heat exchange through the sea surface. So we thought that we're going to give it a shot and look using over 20 years of data on one of these buoys, see how the heat flux behave and what implications that have for the water column. So that's how one of those buoys look. That's actually the neon. Usually they look like they have more stuff in it, but a hurricane went through the buoy, so we lost a solar panel and stuff like that. But it's still standing, so pretty sturdy. So if you look at this, is a coastal ocean monitoring and prediction system. Comes from the Ocean Circulation Lab. Have been maintained several moorings over the last 20 years. Currently, we have 1, 2, 3, 4 real-time data, real-time moorings that send data, obviously, really frequently. And we also have 3 more that are bottom mount, so we don't. There's no real time, but we record it every year. Particularly, we're going to be using these 2, so C10 and C12. So C10 is the main one that we're gonna be doing the heat fluxes, and C12, we try to like fill gaps in the meteorological data when we didn't have available data in C10. This is how the data look. We have a full set of like meteorological sensors, 3 or 4 CTDs on the cable. Plus we have currents, and what makes C10 and special mooring is that it does record incoming short wave and incoming long wave radiation. As far as I know, it's the only like long-term mooring on the East Gulf of Mexico that have a record of that. We need those incoming short wave and long wave radiations to be able to estimate heat fluxes. All right, so like a little bit of the methodology we are using to estimate the heat fluxes. We take all the 7 variables and we use the Coppola Ocean Atmosphere Response Experiment by flux algorithm. It's been around for a while. It's pretty standard. But since my friends were bullying me about like we physicists not explaining stuff, I went ahead and like channeled my inner 5-year-old and make this really pretty flat. So basically, when we're going to compute net heat flux, we have 4 main terms. We have the main heating term is incoming short wave from the sun. So basically, as we all know, radiation from the sun comes and hits the water. It hits the surface of the ocean. Then we have to make a correction for albedo. Some part is reflected back, but most of it makes it back to the inside into the ocean and warms the ocean. And that's the main warming part. And then we have 3 other terms that constitute net losses. First, we have the long wave radiation, which basically is like the surface of the ocean, as it has temperature, is going to emit like infrared radiation, and that's a loss from the surface of the ocean. Some of that radiation is escaped through the atmosphere. Some is absorbed in the atmosphere, in the clouds, and reflected back. So the sum of the loss from the surface of the ocean and the gain from the clouds is the net long wave radiation. The next term is the latent heat flux and the sensible heat flux. The best way to think about sensible heat flux: you turn on your stove, you put your hand on top. That's sensible heat flux. It's the heat you can actually feel. Whereas latent heat flux is the heat that is carried through water vapor changes in phases. So the evaporation requires energy, like it takes the energy as a water vapor. The water vapor like raises, and once it condenses on the clouds, it releases a heat, and that's one of the main mechanisms behind like hurricanes and storms. I think that's it. Oh, and finally we have like the penetrative radiation, which is just part of the incoming radiation. Since this side is only 25 meters depth, some reflects it back in this bottom and makes it out of the ocean. Now that we all know like the components, and my friends I hope are happy. So we went to that data and we computed the heat fluxes for the full 23 years of data. As you can see, it's a little like sparse. There's some like gaps in the data. We need like 7 or 8 variables to compute that. But there's enough data so we can go ahead and try to like take a look at like the seasonality of the heat flux. So for that, we have the plot on the top is the net heat flux. Is the line that the color you should focus is the green one. Is the net heat flux. The other one are the components that we just explained, and the plot on the bottom is surface temperature. So this is monthly mean climatology from the 1998 to 2020. The main point here is like the ocean begins to like gain heat during February, after February, and that heat lasts through August with a maximum in May due to like solar radiation. And if you look at the temperature, the sea surface temperature starts to increase in February and remains like increasing till August, when this net heat flux of 3 signs becomes negative. The ocean begins to release heat and the temperature starts to decrease. And the minimum in net heat flux is during October, because it's a month when we have the confluence of a lot of hurricanes and a lot of cold fronts. So as a main result, we can say that the net heat flux is the main driver of the sea surface temperature, and it's also the main driver on the water color temperature. So this is again monthly mean climatology for C10 and C12. This is a 25 meter depth. This is a 50 meter depth. And we can see like the water column begins to stratify at C10, which is deeper, like at C12, which is deeper like around April, at C10 like around March, and like stratification lasts till like September or October. So the heat flux is still running, like driving the seasonality, but there's like 1 to 2 month lag on the seasonal cycle. But done with like climatologies. Climatologies are, I wouldn't say boring, but they're boring. So now we're gonna like focus on something more interesting. What we know, like the normal state, like what we're supposed to happen with the seasonal cycle. So what are the like main events that disrupt this seasonality? So how we made this a lot of information, but he was talking about like circulation and the loop current. So here we see the loop current for 2010. For the people who don't care about the loop current, probably you have a better life than me. The loop current like move in and out. They go sometimes move west, and sometimes it move east, and it tries this little island here, the little key, the dry tortugas. So when Dr. Liu back in 2016 published a paper and coined this like pressure point concept, which basically is when the loop current interacts with the West Florida shelf on the dry tortugas due to the convergence of the isobats there. Don't get too bored. That sets the whole continental shelf in motion. So this is when that happens. This is the result. This is a bottom circulation snapshot for June of 2010, and what we see is like the color is temperature, and the arrows is the currents, and what we see is like a welling. Like when the loop current touches the pressure point, we have a welling to hold the continental shelf. And then like we thought like, okay, if that happens every time that the loop current interacts with the dry tortugas, can we see that on the data? And of course, like we could. So what we see like the bottom two plots is C10, the other two are C12. 2000 is a year with no influence of offshore of the loop current. 2010 is a year we just saw. And what we can see is that for a normal year, where the seasonality, the seasonal cycle kind of like holds, the temperature difference in between surface and bottom are like maybe like 3 degrees for C10 and 5 degrees for C12, where for like the years where the loop current is bringing these colder waters from the bottom, the temperature differences are between 9 and 10 degrees from surface to bottom. So for the years where the loop current is intruding and bringing this like anomalous weather, like all that seasonality that we just saw goes out of the window. It doesn't hold anymore. And finally, like climatologists can be a little bit subjective, so we try to like kind of like constrain a little bit that. We took the 1D temperature equation, did a really simple like calculation. We used the heat flux seasonality, heat flux seasonal cycle to see how much of the surface temperature seasonal cycle could we reproduce. So the line in red is the one that we are reproducing. The line in white in blue is the actual seasonal cycle. And what we can see is like, obviously they're not gonna match because in this equation, we are not accounting for ocean dynamics, but we can explain the fall, we can reproduce the fall and spring transition and roughly have like the same shape of the curve, which tells us that for this area, that heat flux is the main driver of water temperature and it's not ocean dynamics. With that, so like the main points, the spring transition starts in February and lasts through August, with a maximum heating occurring in May. The fall transition starts in September and lasts through January, with maximum cooling in October. The heat flux drives the sea surface and water column temperature. Sorry. Certification lags surface heating by 1 or 2 months. And the cool part, like the loop current interaction with the West Florida shelf, is the main disruption event to this seasonality of water column temperature. So what's next? This is chapter 1 of my dissertation. After that, like there's plenty of avenues to go to. The more interesting is trying to look the same analysis, but instead of like climatology, it's doing to like different time scale short events, like synoptic scale events, like hurricane cold fronts. I think right now we like picked like 15 hurricanes and storms that went through a buoy. So we're really excited about that. Thank you, and questions. Or you don't have to ask questions.
Just no students asking a question. Oh, go ahead.
I have a question too. So I really liked the point that you're going to compare to the reanalysis products.
Yeah.
Because a lot of people use those reanalysis products to drive models, and in many parts of the graph has been demonstrated. I'm just curious, what reanalysis products are you gonna like?
Oh, like that's like not for this month, so I still don't know.
Yeah,
It's
I
probably for
Biggies or NSAP and,
yeah, like for sure, like NSA, like ERA, something like the European one.
Have you considered looking at the satellite products? Because there are various satellite products of some components of fluxes.
I haven't, but I'm definitely gonna like ask you on that. I'm open to suggestions.
So I think that would be interesting too.
Okay.
Okay.
Yeah.
So going back to the first part that you thought was boring, which I didn't, have you averaged the monthly average of points and just showed one curve?
Yeah.
But what's it look like if you plotted all 12 curves?
Well, it's a mess. It's well, it's not a mess, but it has a lot of variability.
Well, the reason I ask is, it'd be interesting to look at that and compare it to the how warm each year is on the.
I
weather.
didn't have time to show that, but on the paper that comes with that is locally going to be submitted this next week, we see some interesting signals. So the record is not long enough. It's like 22 years to talk about like some climatic signal maybe, but they're like warm and cold phases that you see, and actually like the same reflects on the temperature record in like the airport in Tampa. So there's like I can show a little bit about that.
That's interesting.
Boris.
It's an interesting presentation, and you were looking into climatology of different processes, but what you didn't do, you didn't compute the turbulent fluxes. So, you have fluctuations of temperature and velocity, you can compute correlation, you can compute turbulent fluxes, not only vertical but also horizontal, and they will have big effect on your physics, because by looking at all these processes, it doesn't tell you much about the physics.
Yeah.
Extend it in this direction.
Yeah, of course. Yeah, like I would like ask you about that. Then that would be like a good like closure to that to this part. Thank you. Well, now we're
Thank you, Louise. We're going to take a quick break now. At about 10:55, we're going to do the first raffle. So if you want to be back for that, and then we'll start presentations again at 11.
And before you completely get up from your seats, some of you have lucky chairs, so feel free to take a look under your seat. If there's a like a prize waiting for
Just having enough of each.
Yeah. I better stand up.
Of course. In terms of introducing them, I feel like rather than reading out the profile,